This is part 3 of the Chapter 14 lecture notes, and this is just a picture of Elizabeth Blackburn, who is the 2009 receiver of the Nobel Prize, and she discovered how telomerase works. This is an image that is showing you how what happens when a mutation doesn't get corrected. So uncorrected replication errors. And mutations there are different types of mutations. There are point mutations where just one nucleotide is different, okay? So it's showing you right here, and I guess I should explain this. Every 3 nucleotides in the DNA codes for a specific amino acid, and what you're seeing are the abbreviations of the amino acid names. SER is actually serine, VAL is actually valine, and I won't tell you all of them. I just want you to understand what you're looking at. So every 3 nucleotides in the DNA codes for a particular amino acid. So basically what DNA does is DNA codes for proteins because proteins are made of amino acids. So instead of saying that DNA codes for our traits, we can now say DNA codes for specific proteins, and those proteins do determine our traits. But if you will notice that there is a mutation here, this A was changed to a T. This is a silent mutation because valine actually has more than one code that can code for it. GTA codes for valine and GTT codes for valine. In fact, most of these amino acids have 4 to 6 triplet codes that can code for them, so that some mutations end up being silent mutations. And it's a point mutation because it's just one nucleotide that's been exchanged for another. The A at one point was changed to a T, so that's a point mutation. A missense mutation results when an amino acid is substituted, when it results in a different amino acid. So let's see here. So what we had was proline here, and it looks like an A. Yeah, this is a point mutation where the C becomes an A. And that CCC codes for proline, but that when it becomes ACC, that codes for threonine. Now, that amino acid could cause extreme changes in the protein. That one amino acid could make a huge difference. It could mutate the protein. A nonsense mutation, a nonsense point mutation, is when a substitution actually codes for a stop codon. So here is the amino acid tyrosine, and we're looking at a situation where of course, proteins can have thousands of amino acids, hundreds of thousands of amino acids. So this is just one of them. And we're assuming that there's many thousands more after tyrosine. But if the C is replaced with a G, TAG doesn't code for an amino acid. It codes for stop. It literally means stop making protein. Then the proline would be the very last amino acid in that protein chain. If this happened early, the protein could be very short. If this happened late, then it could just be missing a piece. But it's definitely going to affect the protein. It's going to all of the amino acids after proline will not be attached to that chain. So a nonsense mutation could be a big deal. A missense mutation could be a big deal. silent mutations don't cause any problems. Now, frame shift mutations are normally pretty bad because that means that an amino acid is inserted or deleted, and that just shifts the whole frame, it shifts the whole sequence. So if we started out with serine and valine, and it looks like a T, let's see, AGC, and then G, a T and an A were inserted before the CCC. So these two, in the original, they were there, and then in the new one, they're actually deleted. They are no longer there. So the T and the A have been deleted, and that deletion shifts the frame, and it causes the new amino acids to be serine, valine, which are like the original, but then leucine and leucine, which are not. So, and the same thing could happen if, in this case, we're looking at a deletion, but you could also have an insertion, and that would shift the reading frame, and it would change the amino acid sequences. So this is the same exact. Point mutations can be silent missense or nonsense, and frame shift mutations can be insertions or deletions. Then you can have the entire chromosome mutations. Chromosome mutations mean that a larger piece of the DNA is inserted or deleted. A long piece of the chromosome is inserted or deleted or translocated. It's removed and then it's moved to another location, or it's inverted. Two pieces exchange places. There are also fusions and there are duplications in whole chromosomes. So these are bigger mutations and they usually cause more problems. This is a TED Talk. I like TED Talks, and this goes with slide 24. Now, this is a little bit of a review, but this is a eukaryotic cell versus a prokaryotic cell. A eukaryote contains a well-defined nucleus, whereas a prokaryote doesn't. The chromosome, the single circular chromosome, lies in the nucleoid region. In a eukaryotic cell, the individual chromosomes, which are rod-shaped, they're not circular, lie in the nucleus. And when the cell is not dividing, we call the DNA chromatin because it's very spread out and diffused. So when DNA does compact and form chromosomes, that's going to happen, remember, in prophase of mitosis. So you start out, to understand this DNA compaction, you start out with the DNA double helix, and that's naked DNA, that's just plain DNA. And that DNA wraps around histone proteins. And so now that combination of DNA and histone proteins is called a nucleosome. The nucleosomes are coiled up to form, that should be form, a solenoid shape. My husband says solenoid. He says because it's a part of a car. Don't ask me which part. But anyway, that it forms that shape. And the solenoid is further looped back and forth, attaching to scaffolding proteins. And then the fully compacted DNA and packaging proteins form a condensed chromosome. So this is just the steps in the formation of a chromosome. The nucleosome is the form that the DNA is in whenever it's still chromatin. And then once it compacts, it is called a chromosome. This is an image trying to explain the Sanger method of sequencing DNA. Basically, there are dideoxynucleotides, which are used to generate DNA fragments of different lengths. And we know the nucleotide on the very end. So this one we know ended with a C, and this one ended with an A, and this one ended with a T, and this one ended with a G. And so you just sequence the same piece of DNA, make many, many copies of it, and you just, the different copies will end at different nucleotides. And you always know which one it ended with because you labeled it, you radioactively, or you labeled it with a dye. And so you can tell the length of every one of those pieces, and you know it ended, you know which nucleotide it ended with. So we know a T is here, we know an A is here, we know a G is here, we know a C is here. So electrophoresis, like you are using in your lab this week, is used to separate pieces of DNA based on size. So we know where the smallest pieces are, and we know where the largest pieces are because bands in the gel tell us that. The larger bands are closer to where you loaded the DNA in the wells, and or the negative end, and the smaller bands move further down the gel, and they're located at more at the positive end. So this is the negative end of a gel, and this is the positive end of the gel. And to show you that, this is a gel electrophoresis that uses DNA. Your dye gel electrophoresis is not nearly as impressive, and I apologize for that, but we don't actually use DNA. We use different colors of dyes just to show that the dyes with the different colors, those different pigments are different sizes, kind of like when you did your chromatogram in the photosynthesis lab. So the dye will separate just like DNA will. But we could look at a band, let's say this band versus this band. Let's say that the bright band is A and the lighter band is B. So band A is smaller in size. It is a shorter sized band. I've said it wrong. I'm so sorry. Band A is larger in size. It is actually a larger molecule than band B. Band B is a smaller molecule. So we can use gel electrophoresis to, we know these bands that are brightly colored are the ones, like we will label them with, we know that they end with A or C or T or G. Okay, so if the one that's brightly colored, we know that ended with an A, then this tells us the position, tells us how large the piece that ended with the A is. And so we know where A is, what position it's in the DNA. Okay, and that is it for chapter 14.